All right, today we're going behind the scenes into an atomic level movie studio that is literally changing how we see life itself.
So here's the fundamental problem scientists have been wrestling with for ages. The molecules that make us tick, proteins, DNA, they're not just static little sculptures. Nope. They are constantly moving, vibrating, and dancing in ways that are incredibly fast. This dance is everything. It's how we live. So the big question is, how on earth do you watch it?
You can't. Not with a normal microscope anyway. It's just too small and too fast. But what you can do, and this is where it gets mind-blowing, is build a perfect digital replica of that molecule. Then you use the fundamental laws of physics to hit play and watch it go, frame by frame, femtosecond by femtosecond. And that is molecular dynamics, or MD for short.
So here's our game plan. First, we'll peek into the secret life of these molecules. Then we'll see how scientists actually build these virtual movies. After that, we'll learn to decode the dance, see how these simulations lead to real-world solutions, and finally, we'll look at what the future holds for this incredible tech. Okay, first things first. We really need to appreciate just how wild the molecular world is. It's not some static picture in a textbook. It's a non-stop, high-speed ballet. The thing is, a molecule's job is defined by its movement. I mean, think about an enzyme doing its thing, or a hormone finding its target. These aren't simple, clunky lock and key moments. They are elegant, dynamic dances that happen on the timescale of femtoseconds. And a femtosecond is, get this, a millionth of a billionth of a second. It's an almost impossibly short amount of time. This motion is biology in action, and that's why we need a very special kind of camera to see it.
So how do you actually make one of these movies? How do you go from a 3D model to a full-blown film of a protein in action? Well, it's a process that's kind of like a 3-act play.
Act 1 is initialization. This is where every single atom in our virtual world gets a little random nudge to get it moving. Act 2 is equilibration. This is basically the warm-up act. The system needs some time to settle down from that initial chaos so that things like temperature and pressure can become stable, just like they would in the real world. And finally, Act 3, production. This is the main event. It's when we hit the big red record button and capture the true natural dance of the molecule. And this part is absolutely crucial. Scientists only ever analyze the footage from the production phase. Why? Because that's the only part that shows the molecule behaving naturally, doing its own thing, not just reacting to the artificial push we gave it at the start. This is where the gold, the scientifically valid data is. Okay, so we've shot our movie, we have terabytes of data, basically a film of atoms jiggling around. Now what? How do we turn all that raw footage into something we can actually understand? Well, we use some really clever measurement tools. Our first tool is called RMSD. The easiest way to think about this is as a wobble meter. It tells us how much the molecule's overall shape is changing over time. If the RMSD is low and flat, it means our molecule is stable, holding its form. But if the RMSD is high and all over the place, it means it's unstable. It's wobbling. And here's where it gets really useful. Let's say we have a normal protein, the wild type, and a version with a single mutation. By comparing their wobble meters, we can instantly see the difference. In this case, the mutant protein has a much higher wobble. That tells the scientist right away that this one little change has made the whole protein structurally unstable. It's an amazing way to see the impact of tiny changes. Okay, next up we have RMSF. Now, if RMSD was our wide-angle lens for the whole molecule, RMSF is our zoom lens. It lets us look at each individual part, each amino acid, and see how much it's moving. It's perfect for finding the really flexible bits, the flappy parts and hinges that are often the most important for the protein's job. And our third key tool is the radius of gyration, or RG. This one's pretty simple. It just measures how compact the molecule is. Is it a tight, dense ball of yarn, or is it starting to unravel and get loose? A low RG means it's compact. A high RG means it's expanding. And just like before, we can compare our wild type and mutant proteins. And if we see that the mutant has a consistently higher radius of gyration, that's a huge clue that the mutation is making the protein fall apart and lose its tight structure. You see? These 3 metrics, they turn a chaotic movie into a clear, compelling story. This is all really fascinating stuff, right? But what's the end game? How does watching virtual molecules dance actually help us in the real world? Well, this is where it gets really practical, really fast. One of the absolute biggest applications is in designing new medicines.
See, for a drug to work, it usually has to fit perfectly into a tiny pocket on a target protein, like a key in a lock. Molecular dynamics lets us watch that interaction in super slow motion and then helps us engineer a better key.
You might start with a drug that kind of fits, but it's a bit wobbly. By running these simulations, scientists can see exactly how it moves around in that pocket. They can then make tiny chemical tweaks to the drug's design, run the simulation again, and see if it's a better fit. They do this over and over until they've designed a drug that slots in perfectly and stays there. It's an incredible design tool.
And the level of detail is just astonishing. We can actually count the connections a drug makes and see if it maintains, say, over 80% of them throughout the simulation. We can even spot how individual water molecules can act like tiny little helpers, forming bridges that hold the drug in place. You get the full picture of just how well that drug is working. And it goes way beyond just drug design. Scientists use MD to watch how proteins fold into their complex shapes, to understand exactly how a genetic mutation causes a disease, or even to watch the gates of ion channels in our brain cells open and close. In fact, this technology was a total game changer in developing the first HIV drugs, turning a death sentence into a manageable condition. It's a true Swiss Army knife for modern biology.
So where is all this amazing technology headed? The short answer is pretty simple: longer, bigger, and way more complex.
You know, decades ago, we were thrilled if we could simulate just a few nanoseconds of a molecule's life. But thanks to the explosion in computing power, we've blown past that. We're now deep into the microsecond range and are even starting to reach into milliseconds. That might not sound like a lot, but in the molecular world, that is an absolute eternity. It lets us see much slower, more complicated biological events from start to finish. The ultimate dream is no longer just directing a movie of a single molecule. The new frontier is simulating huge, complex systems. Maybe entire parts of a cell and, maybe one day, a complete, functioning virtual cell. It just leaves you with this incredible thought, doesn't it? We've built this virtual microscope that can see the unseeable dance of life itself. So as this microscope gets more and more powerful, what fundamental mysteries of our own biology, of disease, of consciousness are we finally going to solve?